So for the quiz on Friday, some of the things I'd like you guys to focus on, the things that we covered on Monday and what we'll cover today, includes things like true pathogens versus opportunistic pathogens. Be able to differentiate between those two would be good. I would be familiar with the 5 major factors in the development of infection. So it would be what? The portal of entry, attachment to the host, and then surviving those host defenses, which we'll start talking about today and on Friday, causing disease and damage, and then finally being able to exit the host. We also yeah, sorry, question? Okay. We talked about things like signs versus symptoms, right? Signs are things that can be what? Are the signs objective or subjective? Oh, you're
Objective.
What was that, Brian?
Objective.
Objective. Signs are things, yes, that an observer can measure. Symptoms are going to be subjective. Those are going to be the things that the patient presents with and tells the observer about, but aren't really measurable, right? When someone talks about their pain, that's relative to them. You can't really assess that as well as you can signs like temperature, right? Endogenous agents versus exogenous agents. We talked about that and the differences between the two. One goes into the cell, one comes out of the cell. And then, today we're going to be talking about different lines of defense. We're going to be talking about the immune system. So Monday we talked about the host microbe interactions and how most microbes are beneficial or commensal, meaning they're not mutually benefiting. They are benefiting, but we are not being harmed. And then we talked about some of the tissues that are colonized with microbes, good, bad, and neutral, and tissues that should not be colonized by microbes. And if they are, that leads to, that's an indication of infection. Today, we're going to talk about the first, second, and a little bit about the third lines of defense in the immune system. Okay, so when we are invaded by microbes that aren't beneficial, then the body has to do something to clear those. Okay, so before I open up the slide deck, I do need to quickly plug the Meta Major event that we are having next Tuesday from noon to 1, so October 6th. There's going to be a talk on global climate change, and we have a presenter who has a really nice presentation on that, and then there will be some panelists that will answer questions. And so for those of you students who are enrolled at LTCC that aren't just taking this class from distance, I don't know if it's required, but it's strongly encouraged that you attend. There'll be more information coming out from the PR department with link to the Zoom and stuff. But that's going on. That should be a really interesting talk, especially, right now where we are in it's hard to stick your head in the sand and ignore climate change. So that's happening next Tuesday from noon to 1. All right, let's get into it, shall we? All right. the innate immune response. So chapter 14 is all about the innate immune response. And then on Friday, we'll get into the adaptive immune response, okay? And so they are two different things. We'll talk a little bit about the adaptive immune response toward the end, but primarily today, it's just going to be innate. Okay, so looking at what we'll be talking about, the essential obstacles for microbes. And we talked a little bit about those obstacles being skin. Skin is a great barrier. There's also mucus in our nose to trap microbes. There are defenses like cilia that Ray had asked about last time. What keeps the microbes out of the lungs? The cilia are these, villi type structures that wave and kind of brush it back out to prevent it from going into the lungs. And so in order for a microbe to take hold and infect and do those 5 different factors in the development of infection, they have to get across lots of different barriers. And that's what we'll go into. So innate, non-specific is going to be comprised of first and second lines of defense, and be able to know these, okay? What's a first line of defense? Those are going to be things like physical barriers, skin, mucus, chemical barriers. So like we'll get into that a little bit more, what some of the chemical barriers are, but they're not going to be cells. They're going to be proteins and things like that, enzymes. And then genetic barriers, right? So for instance, if you have sickle cell anemia, your red blood cells don't have the right shape and structure for allowing the malarial parasite to infect. And that's what Plasmodium likes to go. That's the host cell for Plasmodium that causes malaria. It's your red blood cells. And so certain genetic predispositions will make you more likely or less likely to be infected by certain microbes. And then beyond the first line of defense, you have your second line of defense. And this is going to be the inflammatory response or inflammation. It's going to be interferons, which again are not cells, but chemicals that are released to target microbes. Phagocytosis is a cell, a type of cell that then engulfs the microbes and destroys them. And then complement is, again, these proteins. We'll talk a little bit about the proteins, not a whole lot, but again, they're proteins that can either bind a bunch to the outside of a microbe called opsonization. And then as all these proteins cover the outside of that microbe, it's kind of like a red flag or a signal to immune cells to come and get it. So those are the first and second lines of defense. And then the third line is going to be your acquired immune response that we'll talk about on Friday. Your B cells, your T cells, antibodies that specifically bind to microbes and then cause cytotoxicity with your T cells. Okay? We'll talk a little bit about the lineage of different immune cells, where they come from. All of the immune cells come essentially from hematopoietic cells, which are your cells in your bone marrow. And they differentiate into different types of cells with different types of functions, okay? So neutrophils are your phagocytes, the ones that engulf microbes. Your basophils are going to cause inflammation, react to certain allergies you may have. And then your eosinophils are primarily responsible for worm and fungal infections. okay, as well as the inflammation. And as I said, all these immune cells are going to come from hematopoietic cells, blood cells, okay? The bone marrow produces these. And so that is where everything comes from. If something happens to your bone marrow, then you are going to be severely immunocompromised, because that's the origin of all of your immune responses of the cellular type, right? You still have your first line, but that's going to wipe out a lot of your second and third line of defense if your bone marrow isn't functioning properly. And then the wandering search parties. So these cells, these immune cells, aren't stuck. They're not immobilized, right? They're constantly moving throughout your body, whether it's your bloodstream or your lymphatic system. They're moving around throughout your body, looking for things to attack. Okay, and that's how your body surveils these pathogens. And again, if your immune cells aren't getting the right signals, or they're getting confused, then they'll start attacking native tissues, native cells. And as a result, you have an autoimmune disorder. And then we'll kind of go through a little demonstration of what happens when you get injured, right? And again, when you get injured, like stepping on a nail that has microbes on it, then that's going to break your first line of defense, your skin. And then we'll kind of just go through and talk about what happens, how your body responds to that on the different levels, these different lines of defense. We'll talk about phagocytes a little bit in more detail and what they do, how they engulf the pathogen, and then how they use things like lysozymes and other enzymes to break those pathogens down. And remember, I had mentioned before that some bacterial cells and pathogen and other microbial pathogens have adapted ways to evade this breakdown. So they may get engulfed by the phagocyte, but they are resistant to the lysozyme and enzyme degradation. Okay, so just to review, innate natural defenses are going to be non-specific. They're going to be present at birth, right? You're born with a certain repertoire of innate immune defense. Adaptive is specific, and that's what you're going to acquire over time. You will get some maternal antibodies from the mother during development, and if the baby is breastfeeding, but those wear off after a short period of time, and then it's up to the baby to start acquiring an immune response to certain microbes. Okay, any questions before we dive in further? Okay, so here's a nice table breaking it down. Your first line of defense, non-specific, innate. Barriers are going to be things like skin, mucus, tears, right? Your eyes flush out all kinds of microbes just by having tears in your tear ducts. When you sneeze or you cough, you are trying to expel something that shouldn't be there. They used to think that when you sneezed, you were possessed by a demon, and that's why they say, bless you. Now we know that you sneeze not because you are possessed, but because you're trying to get rid of something that shouldn't be there. And so that's why when my students cough and when my kids cough, I nerd out and I don't say bless you, I say congratulations, right? You don't need to be blessed for sneezing. Nothing's bad happening. In fact, it's a good thing. So congrats, you've sneezed. You've gotten rid of something that shouldn't be in your respiratory system. Chemical barriers are going to be things like low pH, right? Your stomach acid is a low pH, and it's going to kill a lot of microbes that can't survive at that in that condition. Lysozymes, which are enzymes, digestive enzymes that break things down. Those are your chemical barriers. And then genetic barriers are going to be things that make you are going to make you incapable of being hospitable to a certain pathogen. And one of the examples I used was the sickle cell anemia, which is not great for the host to have it, but it's evolved in that host because while sickle cell anemia is a disease and it disrupts your ability to carry out normal gas exchange, it's not as detrimental as malaria. So it's evolved in the host because that's more that, they survive longer with the sickle cell than they would with malaria. Second line, phagocytosis, inflammation, fever, and interferon. Okay. Third line, T cells, B cells, and the antibodies that are produced. Be able to know these differences, be able to explain and recognize examples of first, second, and third lines of defense. So the barriers, again, built-in defenses in the skin. Epithelial cells are very strong. The keratin in your skin, also in your nails, in your hair, is enough to keep out most microbes, right? If we sneeze, cough, cry, sweat, that's ways to externally flush out these bacterial and viral and fungal invaders that are waiting to enter. Mucous membranes, great ways to suck up microbes and prevent them from exploring further inside your body. So these are, again, just some of the first lines of defense, and I won't belabor that anymore. So let's go on to the next line. So we have things like, or not the next line, but the other examples of things like your sebaceous secretion. So like, your sebaceous. So like what is it? What am I trying to think of here?
What? Or oil glands?
Oil. I don't know why that was so hard. Yeah. So the oil that comes out of your pores, your earwax, right? That's all there to protect you. And, for most of us, it's an annoyance because if you get too much of that, then it's you look greasy and stuff like that. But there's a purpose that we have those things. And that is the purpose. Defensins. So those defensins are peptides. A peptide is just a chain of, or peptides are a chain of amino acids. Okay, so not necessarily like a whole protein, but a little piece of a protein that can then lyse bacteria and fungi, or break open their cells. And once they break open, they're no longer alive. Lactic acid, electrolyte concentration, pH, all of these things help us kill off microbes from a first line defense approach. There's also the genetic defenses, and I mentioned malaria, but there's also a lot of host specificity. Oh, I just draw something. Where did that line come from? So for instance, viruses have a species-specific infection so that, you can't give mumps to your cat, or your favorite houseplant isn't going to get COVID-19, right? It's very specific to the host. And we talked a little bit about that on Monday, like how, I think Ryan's question of what's a good pet to have from a microbiological standpoint. And, most of these animals have significantly different genetic makeup so that the bugs that infect them are segregated from us. Not always. There's always some, there's a Venn diagram, right? There's crossover, but a lot of this allows us to coexist with other species and not get the things that get them. All right, so let's have a little check here. Which is not an example of the immune system's first line of defense? Anybody?
E.
E. E is an elephant. D is in dog. Oh, I see some chats here. Let's see. No one wants to say it out loud. Okay, I'm seeing B. I'm seeing A. A.
A.
A. Okay.
I think A.
A. Okay. A lot of people say A. Some people say B. Let's think about this. Is sneezing a first line of defense? Yes. Just shout it out. Is sneezing a first line of defense? Yes.
Yes.
Is the cilia first line of defense?
Yes.
The cilia is the stuff that brings the microbes out of your respiratory tract. Yes, it's the first line. Hydrochloric acid in the stomach, is that a first line?
Yes.
Yeah, the chemical barrier, right? Lysozyme, is that a first line of defense?
Yes.
No.
No.
No. All right. The answer is A, because inflammation is what we'll talk about soon as a second line of defense. The lysozyme, remember, lysozymes, right? Right here. Those are going to be chemicals. Where is it here? Right here. A lysozyme is going to be chemicals that break down microbes. Okay, so right here, the lysozymes are in these lysosomes, these little green things, these enzymes, and that's what breaks it down. So that's a first line. Okay. So immunology is a very complicated field. In fact, in grad school, it was the hardest class to take, and it was the hardest. I feel like they had the hardest exams and qualifying exams and things, just because it's so complicated. There's so much going on. It's very complex. So many different pathways, and we are just going to scratch the surface of it in this class. But immunology is a totally separate class, like course onto its own. All right. It involves the second and third lines of defense. Okay. So it's going to be these cells and these chemicals that surveil the body and look for different things that are considered foreign and then attack it. And then with the third line of defense, create a memory for those microbes. And the whole point of that is so that they can attack the microbe more quickly next time, right? And you don't get the infection. White blood cells, those are going to be your leukocytes, okay? These are going to be innate cells that recognize and differentiate any foreign material, okay? They're going to be non-self and self, okay? So sometimes when you have a cell that has mutated, you don't want that cell anymore, right? Talking about cancer on Monday with the Journal Club, your cells mutate. And oftentimes, we don't get a tumor because our immune system has detected that cell was mutated, and it destroys it, okay? It's not that cancer is when someone gets cancer, it's that they've had that one mutation and that's led to it. Our bodies, all of our bodies are having cells that mutate, all the time. And the immune cells keep it in check because these mutated transcripts get actually put out on the surface of the cell. So if you have a cell here and the genome is mutated, it's going to translate a little protein of that mutated sequence, and it's going to spit it out on the wall of the cell like this, like just these little receptors. And then the immune cell comes along, and it's right here, right? And it starts scanning all the receptors on that cell. And if it detects something that is not right, whether that's a foreign cell, like a bacterial cell, or if it's a mutated cell, like they say, oh, that protein's not supposed to be produced by this cell. That is foreign. It will then attack it. Okay? So our immune system is great at not only keeping us from being infected by microbes, but also clearing up bad cells in our body and dead cells in our body, right? They got to go somewhere. PAMPs and PRRs. These are kind of along the lines of the complement that I mentioned, right? In the second line of defense. PAMPs are pathogen-associated patterns, molecules shared by microorganisms that trigger an immune response. Pathogen recognition receptors are receptors on white blood cells that recognize the PAMPs, okay? So let's say this isn't a cancerous mutated cell. Let's say this is a bacterial cell, okay? It's going to have a receptor on it here. All these receptors, those are PAMPs, okay? Those are, or it might have molecules inside the cell too that can be detected that it's excreting, right? Certain toxins, but typically it's going to be something that the white blood cell, using its PRR, its pathogen recognition receptors on its cell, right? It's going to detect that and say, oh, that's foreign. That doesn't belong here. I'm going to get rid of it. Does that make sense? Any questions on that? Okay. Hematopoiesis, that is the production of blood cells. And remember, all the immune cells come from blood cells. They're initially stem cells that then differentiate into the different flavors of immune cells and other cells like your red blood cells. And so this can be broken down into platelets, red blood cells, and leukocytes, or commonly called white blood cells. The leukocytes can then differentiate into two different categories, the granulocytes and the agranulocytes. And so I'm going to work through this, this diagram of the differentiation, because it's important that you know all of these cells, where they come from. It might be important that you know it by the end of this week. So we start off with the hemopoietic stem cell, all right? That's going to be derived from your bone marrow. And it can go one of two ways. It can differentiate into a myeloid stem cell, or it can differentiate into a lymphoid stem cell. Okay. From there, oh darn it. Okay. You're not gonna let me do that, I see. There. From there, a myeloid stem cell can differentiate into three different types: the erythroblast, the megakaryoblast, and the monoblast. Those all come from myeloid stem cells. Lymphoblasts come from the lymphoid stem cell. Okay? Now from there, we have the megakaryoblast going into the megakaryocyte. We also have the myeloid stem cell forming a myeloblast. And what happens here? So these guys, the erythroblast and the megakaryoblast, aren't really much involved in the immune response. Okay, particularly the erythroblasts go to RBCs or erythrocytes, and those are responsible for gas exchange. And then your megakaryocytes form platelets, which are responsible for clotting, right? Some degree of inflammation, so a little bit of the immune response for megakaryocytes. Your myeloblasts are then going to form the granulocytes. Okay, so remember I said that there are agranulocytes and granulocytes. The granulocytes come from the myeloblast, and those are going to be your neutrophils, your basophils, your eosinophils that we talked about. Okay, anybody know what line of defense this is? The granulocytes? You have 3 guesses. First, second, or third?
Second.
Second, yes. It's not your first, okay? If it's a cell, it's not really going to be a first line of defense, okay? If it is a cell, it's going to be either second or third. And these don't form memory. These are not part of the adaptive immune response, so they're got to be the second line. These are your phagocytes, okay? These are your cells that release certain cytokines and chemokines that lead to inflammation. And we'll talk about inflammation later. And mast cells, okay? After that, we have now the lymphoblasts differentiating into the agranulocytes. And you can think of this as agranular because, for one, it doesn't have, if you look at these cells under a microscope, you don't see these large granules present in the neutrophils, basophils, and eosinophils. So that's why I believe that's where the name comes from. And these are going to be your B cells and your T cells, your lymphocytes, okay? And that is going to be your third line of defense because that is what's adaptive. The T cells are going to kill the kill the microbial cell. The B cells are going to form the antibodies to recognize it if it tries to come back. But to make things more complicated, right? There's a little bit of crossover. So the myeloid stem cell can differentiate into a monoblast, which then turns into monocytes, which are agranulocytes. Those go on to form macrophages. Macrophages are phagocytic, so they're going to engulf. And also dendritic cells, kind of like macrophages. But they are, again, part of the second line of defense, right? Also, the lymphoblasts go into T and B cells, which are part of the third line, but a lymphoid stem cell can also differentiate into a natural killer cell, okay? That is second line. It's non-specific, okay? If it's specific, it's third line. If it's non-specific, meaning it's just going to go after anything that it recognizes as foreign, then it's second line. The B cells and the T cells, they are specific because once they have a memory of a particular pathogen, they go after those. They create specific cells to go after those microbes. Any questions on this? I know it's kind of a lot, but you need to know this. So if anything is unclear, speak now.
Are the macrophages also granulocytes?
Agranulocytes, yes.
Okay.
So what you can do is draw a line down here, okay? Everything to the right is agranulocyte. Everything to the left is granulocyte. Okay? And so, you know, what we can do is we can, if you want to come up with a table that's better than this, or if you want to come up with some kind of flow chart that works better than this, it might be helpful to look at, break it down to things like, you know, host defense. Does that look backwards to you guys?
No.
No, it looks the right way? Okay, it just looks backwards to me. And then break it down into innate, right? That's going to be your non-specific. From innate, it goes to first line and second line. Sorry if this is kind of hard to see. Can you guys even read this? Maybe. And then, yes.
Says sharing your screen.
What's that? Oh, you can't see this?
Yeah, since you're sharing your screen, it only shows up on a chat.
Small. All right.
If you double click on the screen, it should populate your entire screen.
Yeah. How about that? Is that better? Is that backwards?
It's fine now.
Okay. So you know, at the top, you have all of your defense system. It goes to innate, which is your first and second. And what's in the first line and what's in the second line? Fill that in, right? First line, coughing, sneezing, skin, tears, cilia, so forth. Lysosomes. What's in your second line? Well, we'll talk about that a little bit more, but phagocytes, right? The complement, you know, the PRRs that recognize the PAMPs. Your inflammatory response, that kind of thing. And then what's over here, adaptive. And that's going to be your third line of defense, right? And that's your B cells and your T cells. So it's important that you know these cells and the lineage, but also whether or not they're first, second, or third line of defense. And if you have your chart, you know, if you have a diagram, then you'll know that these really aren't first, second, or third. Platelets, you could say, well, it's involved in inflammation, so it has a small role. Maybe we'll call that second. But red blood cells are not immune cells at all. All of these, whoops, don't go to the monoblast here. All of these are what? First, second, or third? Second.
First. Oh,
Second. Second, because they're non-specific. They attack everyone. This is also second. Non-specific. These are also second. Non-specific. The only thing that is third is this, T cells and B cells. Okay?
I have a quick question. On our exams, would you ever have us like have to like fill in the chart or anything like that? Or we just need to know the information?
Yeah, you could, you know, so in the past, I've had students have to write this out for me. Now that's hard because this is all going to be online. So I don't think I'll have you necessarily write it all out, but maybe I'll have something like this and you'll have to fill in the cells, right? So maybe this stuff will be crossed out like that, and you'll have to know what they are. Or maybe I'll just say, what type of cells are derived from lymphoid stem cells, and what line of defense do they serve? And so you could say, well, a lymphoid stem cell is going to differentiate into T and B cells, and those are the third line, as well as natural killer cells, and that is the second line. Follow me?
Yes, thank you.
All right. And so it does, like I said, immunology is complicated, and this may seem like it's challenging, but in the grand scheme of things, this is really just the superficial delving into the whole immune response. Okay. Neutrophils, eosinophils, basophils. These are the differences. We've already talked about them. Lymphocytes, you're going to have your B cells, your T cells. We'll talk about those more on Friday. Your monocytes, right? Those are going to be your macrophages, your dendritic cells. Those are going to be second line, non-specific. So that brings us to second line of defense. That's going to be the inflammatory response. They have to recognize the pathogen. They have to initiate the inflammation response, and they need to either engulf them or secrete what's called interferon, which is an innate response to a lot of viral infections, particularly. They're non-specific, right? And it can be really, once you've broken through your first line of defense with your skin, right? You get an injury. They're going to be the ones that come to that injured site and start initiating an immune response, right? And that's why if you get cut, it gets kind of red on the outside of that laceration, right? It's being inflamed. I don't want to use the first line, but it's the guys that are coming in before your third line of defense, right? Those guys have to come in afterward to clean it up and to now remember the microbes that it has encountered. This is going to be defined by redness, right? If something's inflamed, it's red. Why is it red? Well, because your blood vessels are dilating to allow increased circulation. Why? To get all of your secondary and third line of defense to that site. It's going to be sometimes warm, okay, because of the increased blood flow. And there's going to be swelling because you are now going to have your lymphatic system going there to bring the immune cells to that region. You're going to have a bunch of microbes there, of course, that are trying to reproduce, and then all the cells that are there destroying those microbes. And as the result of that destruction, you get all this debris and all of that fluid, all of the dead skin cells, the dead microbial cells, and the white blood cells are what form that pus. That's what, when you have an infected cut, that pus is all of that stuff combined, okay? And of course, pain, right? Inflammation hurts because not only are these inflammatory agents, harmful to the pathogen, but they're kind of discomforting to us as well. In fact, when you have that second response coming, it's called often a cytokine storm when you first get infected, particularly with viruses, and your body just hurts, right? You get the flu really badly, you feel like you've been hit by a truck. And that's because you have this wave of toxic chemicals in your body that have been produced and secreted to destroy that virus. All right. So let's walk through. Yeah. What's up?
So like when your body makes like whiteheads for acne, is it kind of like the same concept of that?
Yeah, so that's going to be all of that. It's going to be your sebum, it's going to be the microbes, and it's going to be the immune cells all trapped in that pore, and it can't get out. So it just walls it off, and then you get a pimple, you get a whitehead. It's essentially oil and dead cells that can't get out.
But isn't it like it's bacteria trapped in your pore and it's your body trying to not let it spread, so it's pushing it out?
Yeah, because remember, microbes are ubiquitous and they get into your pores, and your body then tries to prevent that. And if you have a bunch of oil there and it clogs the pore, it can't flush it out, so it just stays there. But yeah, acne is a result of overproduction of oil and in response to trying to fend off microbes. So let's say you step on a nail, which I've stepped on a nail. It hurts, right? And now you've punctured your first line of defense, being your skin. And let's say you've gone into a blood vessel as well. What happens? Well, first of all, you have vasoconstriction of that injured blood vessel, right? So that you don't hemorrhage and bleed to death. Your platelets come to clot the injured blood vessel. But that nail has microbes, it has bacteria on it. And now that bacteria has entered inside your body via that nail. You're going to start getting a flush of these mast cells that are just hanging out underneath your epithelium, right? Just there. Kind of like the National Guard, right? They're just waiting for something like this to happen. And when it happens, they release chemokines and cytokines, these chemicals, into the injured area. And that is your second line of defense. Okay, it's going to help control those bacterial cells from just going hog wild and proliferating like crazy before your third line can come in and your other second line defenses. So you get subsequent vascular reaction. So there was the vasoconstriction to cut off bleeding, but now there's going to be the inflammation response. So vasodilation, the blood vessels around that injury are going to get bigger to flood it with your second line defense mechanisms. All right. There will be some leakage of plasma from the actual, so a blood vessel is not like a hose per se. It's a bunch of cells together. And so if you look here, I don't know if you guys have had anatomy or not, or taking it now, but that is one cell, and that is one cell. And in between is where those two cells articulate, and things can come out or things can come in. It's called intravasate or extravasate. And so things start leaking out of these out of these vessels, including your neutrophils and eventually your white blood cells, right? Your third line of defense to then attack that bacteria. After a while, you start getting edematous, right? Edema, you swell, pus is formed, because now you have all these players here undergoing a microbial combat. And as a result, this is like the battlefield, right? And then you have all the fallen soldiers laying about, and that is what pus is, okay? We have a fibrous exudate. That is just, remember I mentioned, I think on Monday, that you can form granulomas, and it's the body's way of walling off a particular area and surrounding it so that the microbes can't pass further out into other places of the body. So it starts laying down these collagenous fibers around that injured site to prevent the bacteria that aren't being currently destroyed from moving on to other places. It's really a cool, I mean, I find that, the fact that the body does this is such a cool thing. I mean, the response to this is just amazing. I heard some screaming. I don't hear any more screaming. Okay. And then your macrophages, your lymphocytes, fibroblasts come in. The fibroblasts are there to repair the injury. The macrophages are there to clean up everything, to eat up all the dead cells and get rid of the pus and stuff like that. And then your lymphocytes are there to then remember these guys, these bad guys. So the next time they come, the infection won't be able to mount, right? Because they'll be waiting. They'll already be primed and ready. Okay. It is now 10:50. Alas, I did not make it through the end of this slide deck, but we'll pick up on Friday where we left off. There is, shouldn't be too many more slides after that, but, oh yeah, there are only about 5 or 6 slides left. So everything up to here, up to this point that I've covered, is fair game for the quiz, all right, on Friday. So focus again on things that we talked about on Monday: true pathogens, opportunistic pathogens, the 5 major ways that microbes set up infection, signs versus symptoms, first line versus second line versus third line, that kind of stuff, okay? Any questions? If not, feel free to sign off. Use this time. What is today? Today is the Labster. There's just one Labster today, okay? So if you haven't already done that, do that. If you've already done that, or there's only one, so if you have time, use this lab period now to think about your topic. Your topic is due Friday. Now, there's also no lab Friday except for using that time period if you wish to come up with your topic, okay? So don't delay on that. Work on that. If there are any questions, I'm happy to stick around for a few minutes and address that. If not, I'll see you Friday.
Professor, is the Labster up yet?
I believe it is. Oh,
It is.
awesome. What?
I was saying I was able to access it yesterday, so it is up.
Cell culture. I wanted you guys to do cell culture so you could appreciate the immortalized stem cells that came from Henrietta Lacks, and what we do with cell culture. It's not trivial. Also, I failed to mention that there is a video that I put up in the research proposal folder or page. Just a brief overview on going through that template and what how you can use that to help drive your project. So take a look at that video if you want, if you're still kind of unsure about this research proposal and what it's about, then I recommend you go there and check that out.
And Professor, what's the time for the quiz next Friday?
Yes, 12 o'clock, noon. So we have the quiz at the beginning, right before lecture, okay? And the topic is just due by the end of Friday, okay? So you can use that lab period in the morning to work on your topic, and then, or, study for the quiz, that kind of thing.
Have we posted Journal Club already for this week?
No, let's see, what is next week? Is there a journal club for next week? I lose track here, let's see.
And
Yes, there is a reading. Oh, cool. So next week we talk about the Tuskegee Syphilis Project, which was, that was definitely unethical, and people, suffered as a result. So yeah, I will post that. It's all ready to go. I'll post that today. And then what else? You were gonna say something else?
Yeah, where I was having some trouble finding the research proposal video on the page. Where do I
Let,
go for that?
let me show you. All right, so let's do student view. If you go to pages,
Okay.
view all pages, you'll have research proposal.
Okay.
And it has the guidelines. Remember, you can work with a partner, one other person jointly, and turn in one proposal. So if you want to do that, pick someone quickly. Here's the template that you'll use, and it's the same one from Chem 103. So, and here's the overview. Here's the video that I did. And then deadlines for certain things that are due.
So do you just want us to email that to you? Or
The topic,
yeah, I'm missing
I will
it.
I'll create an assignment. I'll just create, like, it'll just be type in your topic. Okay. And again, I'm not looking for something robust. It's just, what do you want to work on? Because I got to keep you on track, right? Not you particularly. I just have to we need to be on track so that we don't delay. So I'll just make an assignment. And it'll be due the end of Friday.